Alrighty, guys, I want to talk about bacterial genetics today. And I am going to just focus on where bacteria get their genetic diversity and a little bit about operons and how they regulate uh, their gene expression and things of that nature. Okay, so first of all, a couple of just some facts about bacteria. And again, they have a really short generation time, about 20 minutes for like E. coli in your gut. So every 20 minutes, they are going to be replicating. They adapt very, very quickly, and they adapt between basically two different ways. Evolution via natural selection, meaning that because and I want to make this point clear, you as an individual, or a bacteria as an individual, or a blue whale as an individual do not evolve. Your DNA is kind of set when sperm met egg, or when bacterial cell split in half. You do not evolve. What does evolve from some of my ninth graders that current 10th graders might remember from my class? Anybody remember? The individual does not evolve. A little different than the species, the population. And again, what's the difference between a species and a population? Population is a group of individuals of the same species that have the potential to interbreed. So you can have multiple populations of the same species. They could be separated by both time and distance. Okay, but the population evolves. That is your unit of evolution. The individuals do not. Okay, so bacteria, because they divide every 20 minutes, mistakes happen in the replication of the DNA. Those are mutations. Mutations are the raw material for evolution. Okay, mutations are the raw material. That is one way by which, actually, the only way by which new alleles can come into a population is via mutation. Okay, I should say that is not the only way. That's the only way new alleles can arise in a species. Okay, is via mutation. Now, new alleles can arrive in a population if a member from a different population comes in, and we'll get all into that when we talk about evolution in our next unit. But because bacteria replicate so quickly, mutations happen. If an individual gets a beneficial mutation, one that has a selective advantage in a particular environment, that bacteria is going to replicate at a faster rate and survive at a higher rate than its ones who don't have that mutation. And therefore, that mutation becomes dominant in that population, just like we're seeing with the coronavirus. As it mutates, those mutations that allow that virus to replicate and be transferred to new individuals at a higher rate are the ones that become more dominant in the population, in the people that we're seeing, like the British variant that's currently going right now. Now, also, that individual bacteria can regulate their gene expression and regulate enzyme activity, and so that's a physiological change that they can do. Okay, so just like us, they have a double-stranded DNA molecule, same bases, A, T, C's, and G's. They undergo the process of replication. They also undergo the processes of transcription and translation, basically the same way. There's no RNA processing, things of that nature, but basically the same way. We can take a human gene and put it into a bacterial cell, and that bacterial cell will make the human gene product, i.e., the protein. That's how we make insulin. That's how we make human growth hormone. We use bacteria to make them. Why? Because the process is the same. Why is the process the same? Common origin. Okay? So, um, the bacterial genome, however, is only 4.6 million nucleotides, okay, or base pairs. Ours is 3 billion. So the bacterial genome is really small. But remember, the bacterial genome does not have introns, it only has exons. So it's very efficient and space compact. They got 4,300 genes in an E. coli bacteria. 4,300. We have about 22,000. So even though we have so much more DNA, we still have the number of genes we have is not really all that much greater. Okay, 5 times greater. But their DNA is so much smaller. Okay. So, anyways, in addition to bacterial genomes, so their chromosome, they do have additional small, what we call extra chromosomal chunks of DNA called plasmids. These are small, simple, circular chunks of DNA, like it's a little plasmid, that has only a handful of genes on it 5, 10, 15 genes. They're really small. Okay, and we'll get into that in a sec. We know that bacterial cells replicate by binary fission. They have a single point of origin. There's no worry about telomeres or anything of that nature because it's a complete circle. There's no overlapping ends at all. So they don't have telomeres. That's another difference that we see with bacterial genomes. No telomeres, no histone proteins, um, no introns. Those are all differences that we see, plus that they're much smaller. So a couple of things. As I said earlier, they reproduce really quickly. 20 minutes for E. coli. Okay, so you can get close to a billion bacteria, a couple hundred million, in 12 hours, starting with one. Okay, so again, like I say up here, in one day, you're going to reproduce or replace 20 billion bacteria that you lose each day when you have your morning constitution. Okay? So your body will replace those so quickly because they just replicate so fast. So here's the cool stuff. Through binary fission, most of the bacteria in the colony are genetically identical to the parent cell. If you know anything about bacteria, if you've done, how many of you have worked with bacterial plates, like where you put some bacteria on a little plate and stuff and let it watch it grow? Some of you guys have. Normally, we would have done that at this point because we would be doing a lab. It just hasn't arrived yet. But when we do this particular lab, you will see that when you plate your bacteria, you won't see anything on your agar plates. Just like, I don't see anything. Put it in the incubator, you come back the next day, and you'll see these kind of spots, and some of them might be small and some of them might be large. Those are colonies. Each colony started from one bacteria, and that colony of bacteria that you see are all genetic clones of each other. Okay? So when you sample bacteria, you'll take your little inoculating loop, you'll scrape a colony off, and there may be hundreds of thousands, if not millions, of bacteria in that little scrape that you took, and they're all genetically pretty much identical. However, spontaneous mutation rate of E. coli is 1 times 10 to the minus 7th mutations per gene per cell division. So there's 4,300 genes. So the chance of a mutation occurring in any gene is really low, right? It's really low. It's not very high. However, because you have hundreds of billions of these bacteria in your gut, every single gene in there's going to be at least one bacteria in your gut that has a mutation in every single not there'll be at least one mutation in every single gene in somewhere in your gut, and actually there'll be 2,000 of them on average. So what I'm saying here is that because these bacteria are reproducing so quickly, mutations are happening, they can rapidly change, okay, according to if a mutation is beneficial. All right, then they can rapidly take over your gut. How many of you guys have traveled someplace, ate some food because you're in a completely different environment and you're kind of ill after for the first couple of days of traveling? Okay, yeah. Why? Because you're changing your diet, and that can even happen if you change your diet. You go, let's say you start going taking a you start you take a what do you call that? Low carbohydrate diet, high protein diet type thing. If you switch to something like that, you might feel ill for a while because your bacterial your microbiota that's in your gut has to adapt, it has to change. And it does that through this process of mutation and natural selection and stuff in that. That takes a while sometimes. It can take a couple of days or so on until your gut adjusts to that new environment, the new foods that you are taking in. Okay, so mutation is, like I said, is the mother of all new alleles in a new alleles, not and again, both prokaryotes and eukaryotes undergo mutation. However, prokaryotes can also undergo these three means by which they can increase genetic diversity. Now, we, as sexually reproducing organisms, can increase genetic diversity through the process of meiosis, crossing over, independent assortment, and then random fertilization and mate choice. Okay, that's how we increase our genetic diversity. Bacteria don't quite do that, but they do have these three processes: transformation, transduction, and conjugation. Okay, so what they can do. So here's a simple experiment looking at the impact of recombination between two different strains of bacteria. We start off with a mutant strain that can make the amino acid arginine, but it cannot make the amino acid tryptophan. Bacteria, just like us, need those 20 amino acids. And if they can't make one of them, they're not going to survive or grow and reproduce on a plate, an agar plate that does not have additional nutrients in it. It's what we call a minimal medium plate. Okay, there's no additional nutrients in this, just enough to let the bacteria live, but they can't reproduce because these guys cannot make tryptophan. Well, over here, I got a different strain. It can't make arginine, but it can make tryptophan. They don't grow on a plate. However, if I put the two together, they're different. They can be the same species. They're not. They do not undergo sexual reproduction. But when I plate these two together, all of a sudden I start to see colonies on there that are able to grow. Well, how did that happen? Because they don't sexually reproduce. How do they exchange genetic information? Well, they do it through one of two processes in this particular case. In this particular case, it would either be conjugation or transformation. Okay, and it's going to depend upon the situation. So let's look at what those transformation and transduction, what they are. Let's start off with transformation. Okay, transformation is basically the uptake of naked DNA that's in the environment into a new cell. Naked DNA, what's that mean? When a bacterial cell dies, it releases its DNA, and more importantly, it releases its plasmids into the environment. Well, we now know that just about any species of bacteria can pick up that DNA or those plasmids from the environment and take them in. This was similar to Griffith's experiment. Remember when he had Staphylococcus? He had the smooth strain and the rough strain, and he mixed them together. He heat killed the smooth strain, and he mixed that with the rough strain, injected them into the mice, and the mice died, and so on and so on. He's the guy who discovered that process of transformation. Just to recap, remember the rough strain, which did not have the capsule around it, was not pathogenic. The smooth strain, which had a little capsule around it, was pathogenic. And he heat killed when he injected the mouse with the rough strain, the mouse lived. He injected the mouse with the smooth strain, the mouse died. He heat killed the smooth strain, injected that in, the mouse died. The mouse lived. But when he mixed the heat killed smooth and the rough strain together and injected that into the mouse died. And when he pulled the blood and looked at the blood, he found the living smooth strain once again. He's like, oh, what the heck happened there? And he called that the transformation factor. He didn't really know. But now we know that when those heat-killed cells were mixed with the living rough cells, their DNA was taken up by the living rough cells, and that transformed them into those smooth cells. So that's that uptake of DNA from the environment. Okay, transduction occurs when a bacteriophage, a virus, injects its DNA into a cell. And if you remember from that earlier, the presentation I did on viruses, the bacterial DNA becomes the viral DNA becomes part of the bacterial genome in what we call a prophage, and that lysogenic cycle. And when that virus says, okay, hey, time to replicate more viruses, it creates its DNA, but also takes a little bit of the host cell's DNA, okay, in with it. And when that bacteriophage goes and infects another cell, it can actually put the DNA from one bacteria into another bacterial cell. So it's a movement of DNA, chunks of DNA via viruses. And this is what we do with a lot of gene therapy now, too. We're using viruses, especially the cold virus, to deliver functional genes to people who are immunosuppressant or something of that nature. So we're using that now as a tool. Conjugation is a little bit different, okay? Conjugation has to deal with the plasmids. And basically, what happens is one cell who has the plasmid recognizes a cell that doesn't have the plasmid, and then what's called a sex pilus or a conjugation bridge, which is basically just an extension of the membrane, will form between the cells. And a plasmid will be transferred from a positive cell, one that has a plasmid, to a negative cell, one that doesn't have it. So here's basically what happens is we have the plasmid in this cell, and here's the bacterial chromosome. Of course, this is huge compared to what it really would be if you're really small. When the F plus cell recognizes an F minus cell, a mating bridge, sex pilus, conjugation bridge will form between the two. And then the plasmid will actually replicate into that new cell. So now that bacterial cell has that plasmid. Well, what's the big deal with that? Those plasmids can have genes for like antibiotic resistance. Okay, this is, or they could have genes for breaking down a new food source, something of that nature. That they don't have many genes, but these are genes that can be moved from cell to cell to cell to cell. Okay, so plasmids, and normally I'm actually taking out a bunch of the stuff on the plasmids from the test because we didn't have a chance to do that lab, and that's when I go really into it. So that's the reason why I'm kind of rewriting a bunch of it right now. But we will do that lab once it comes in. Okay, so I'm not going to go into transposons because I don't think that's not in the AP stuff anymore. All right, so let's look at control of gene expression. This is what you guys were working on yesterday with the operon models. Okay, so again, bacteria can cope with changing environmental fluctuations. Okay, two different ways. Actually, three different ways. They can go use natural selection and whatnot, but they can also vary the number of specific enzymes. What kind of enzymes are being produced by regulating gene expression? Like we saw with the POGO you guys were doing the other day. If there's no lactose in the environment, why make the genes to break down lactose if there's no lactose in the environment? Okay, so that's regulating gene expression. Or they can adjust the activity of enzymes. Okay, they can use allosteric inhibition maybe and shut the enzyme down so it doesn't do its job, so to speak. And we'll look at that in a sec right here in our next one. So two different ways: regulate gene expression or change the activity of an enzyme that's already present in cytoplasm. Okay, so create new proteins or adjust the activity of existing ones. All right, so tryptophan. This was the Model 2 that you looked at in your POGO the other day. Okay, tryptophan synthesis. This is an repressible operon. Okay, it's generally going to be on all the time. And what is the tryptophan biosynthesis pathway? It basically takes an amino acid and through a series of 5 enzymatic controlled steps turns it into the amino acid tryptophan. Okay, so we start with one amino acid and turn it into another one, tryptophan at this point. Tryptophan just is necessary for bacteria to live, just as it is necessary for us to live. Okay, once again, why produce tryptophan if you don't need it? If there's already a lot of it in the environment, there's no need to produce it. So through the process, what kind of feedback would this be? Positive or negative feedback? We got the product turning off the synthesis. That's going to be negative. Remember, with negative feedback, the product of any system turns off the system. With positive feedback, the product of the pathway is going to increase the production of more of that. So it keeps on getting more and more and more and more and more. Negative feedback, when you get reaching up, it turns it off. Okay, like think like a heat thermostat. That's negative feedback system. Okay. All right. So um, again, if tryptophan levels are high, tryptophan is going to act as a allosteric inhibitor. It's going to bind to early on in that enzymatic pathway and shut that enzyme down. If you don't have the enzyme one working, you're not going to do the rest. You saved a bunch of energy because there's already plenty of tryptophan in the environment. Why make more? Why waste energy? Okay. So tryptophan can also not only block the enzyme, but it can also shut down gene transcription. All right, so two different things you can do there. Okay, and this is where we get into the opera. Two guys, don't really need to know their names, Francis Jacob and Jacques Monod, but Jacques Monod was a crazy wild man. He was a member of the French resistance during World War II, did all sorts of suicide missions, but survived them all, and ended up becoming a PhD student in California at Berkeley, working under Thomas Hunt Morgan, who was the guy who did all the early fruit fly stuff in the 1900s at Columbia University. So, pretty cool story behind this guy. But, anyways, he discovered the operon system. And again, operons all have three elements. They have the genes that it's going to be turned on or turned off, promoter region, which is where RNA polymerase binds to, and of course the operator region. That's where the regulator protein can bind to. And remember, the operator is going to be the on-off switch. Okay, the operator is the on-off switch. Um, I always like to remember the easy thing that makes me remember it is frog. All right, it's not really in the order, but it makes me remember all the parts of an operon. Promoter, regulatory gene. I'm not spelling these right, but that's okay. Operator, and the genes. Okay? So just remember prog, kind of sounds like frog, easy to remember. And you'll remember all the parts of an opera. Okay? So again, this is our trip. This was model 2 in your book. Okay, excuse me, on your POGL. This was model 2, the trip operon. And remember the trip operon, the regulatory protein is produced in its inactive form. And so this particular operon is always on for the, unless there's a whole bunch of tryptophan in the system. Why is it always on? Because these are the tryptophans essential for that particular cell to survive. It needs tryptophan. So it's always going to make it unless there's plenty that already exists in the environment. Then it will shut it off. Okay? So again, when tryptophan is in the system, it'll bind to the inactive repressor, changing it to an active repressor, which now binds to that operator and turns the system off so we don't make tryptophan. We're not going to waste energy. So that's a repressible operon. It's always on normally, but if tryptophan is, if the product that this operon is going to make is already present, it gets turned off through this active repressor. Okay, I'm going to skip over that. The lac operon, which is model 1 that you looked at in your POGL yesterday, is a little bit different. It's an inducible operon. So when the regulatory gene is transcribed and translated, the protein is already in its active shape. It binds to the operator and turns off that particular system. Okay, I'm going to stop right there and let you guys take your break, and we'll come back in a sec. Let me get back up to here. All right, and let's go to present. All right, so like I said, the lac operon is an example of an inducible operon. This one is always off generally because it's producing enzymes that are going to break down a what I call an ephemeral food source. Anybody know what the term ephemeral means? Means not there all the time, just kind of comes and goes. Okay, you take ecology, you'll learn about ephemeral pools, which are like water sources that kind of dry up during certain times of the years and things like that. And they have really cool ecology about them. Anyways, so these will only this series, this operon is only turned on when there's a certain thing in the environment that's going to bind to the regulatory protein, and then it's going to cause that change, that shape change, which is going to cause that regulatory protein to fall off, and then we can transcribe those genes. So again, in this particular case, the lac operon is called the lac operon because it's talking about the production of the enzymes that are going to break down lactose. We need to have lactose in the environment. Now, they have allolactose. That's just an isomer of lactose, okay? But that's the one that will bind to the repressor protein, causing it to change shape, therefore fall off, allow RNA polymerase to now transcribe and then translate those genes into these three enzymes: beta galactidase, permease, and transacetylase. Okay, you don't need to know those names or anything of that nature, but just recognize they are enzymes, and they're going to basically make the enzymes that are going to break lactose down into glucose and fructose. Okay, so that's what they do. All right, so being able to compare and contrast the two operons. Okay, so as I said up here, repressible operons generally function in anabolic pathways, ones that build things. Okay, synthesizing end products, and these guys are generally on. In the trip operon, they're synthesizing tryptophan, an essential amino acid. Inducible operons usually function in catabolic pathways, digesting nutrients to simpler molecules, such as the lac operon. They are generally off. Both of these operons, these were off, this was in your POGL, demonstrate what we call negative control because the active repressors turn off the operon. When the repressor is active, it's bound to the operator, and then we're not going to make those enzymes in both cases. Okay. Once again, here's the repressor bound to. It's active. It's not. It's going to turn off the operon. Here's the repressor. It is active. It's turning off the operon. Negative control. Okay. The last part of your POGL was Model 3, and that was talking about positive gene control. Okay. So what's going on in positive gene control is your systems are a little bit more complex, and lactose is a disaccharide composed of the monosaccharides of, I think it's glucose and galactose are the two ones that I always forget which ones, but it's two monosaccharides that make up lactose. Okay, well, the bacteria really like glucose more than they like lactose because it's simpler to break down. It's already broken down into a monosaccharide. It does not have to expend energy to break down lactose. So if there's lots of glucose in the system, why bother making these enzymes if there's plenty of food available in a simpler, more easily digested form? Again, saving energy. So there's another additional control system here. Okay, so in E. coli, when glucose is scarce, okay, when there's no glucose and there's lots of lactose in the system, first of all, lactose is going to bind to the operator, bind to the repressor protein, change its shape, and make it fall off. If glucose is low in the system, then another protein is going to become activated. It binds to the promoter region and it enhances polymerase, RNA polymerase. It says, hey, you need to bind right now. Okay? And when this is off and this one is bound, it's going to transcribe those genes in order to break down lactose because there's no other food source available. Okay? So it's going to break down lactose. However, if glucose levels are high and lactose levels are high, all right, remember, if glucose levels are high, it doesn't want to make the lactase genes, because it already has plenty of food available in glucose. But because lactose is high, this repressor protein has fallen off because lactose is bound to it, changes shape. But this cat protein is inactive. It doesn't have that little molecule, which is cyclic AMP. Okay, it's not bound to it. So it's not bound to the promoter region. If it's not bound to the promoter region, RNA polymerase will not bind to it. It just won't. Okay, even though there's nothing blocking transcription here, but because this protein is missing, okay, it will not bind. We call this positive control because when this protein is in its active form, it has a positive effect on the outcome. It's going to transcribe those genes. Okay, but so that's the reason why it's called positive control. Well, that's what I wanted to talk to you about, bacterial genetics. What do you need to know? You need to know, how do bacteria generate genetic diversity? Mutation, we see that in all organisms. Prokaryotes, eukaryotes, everything. Okay? Mutation, viruses also. But bacteria can also generate genetic diversity through the processes of transduction, which is the uptake or the transfer of genes from one bacteria to another by a virus. They can also generate genetic diversity through transformation, where they uptake DNA from the environment, generally in the form of plasmids and things of that nature, or through the process of conjugation, where one bacteria that has a plasmid basically gives it to another bacteria that doesn't have a plasmid. And what's interesting with transformation, any species can grab any other bacterial DNA. Conjugation generally is going to be between the same species of bacteria. But transformation can be between just about anything. So places where there's lots of bacteria and there's lots of selection for, let's say, antibiotic resistance, that might be like a hospital, okay? Lots of bacteria everywhere, okay? But hospitals use a lot of chemicals to try and get the bacterial numbers down, right? They're cleaning with all sorts of antimicrobials, antibacterials, use all sorts of different types of soaps and cleaners and sprays and everything to try and make it clean. Well, that's a selection pressure, guys. That's something in the environment that bacteria are like, oh, we can't get there, but mutations happen. All of a sudden, one is able to withstand that new changed environment. That's the one that survives and reproduces, and now has that genetic antibacterial resistance that it passes on to the next generation. Okay, one of the reasons why, have you guys ever heard of the hygiene hypothesis? Basically, one of the things we think that we see so many allergies now is because we're too clean. We don't stress our immune systems enough. We don't expose them to enough, um, dirt, if you will. Okay, and because of that, when we do become exposed to it, we have a tendency to become overreactive. Our systems overreact. So you can look it up. It's kind of cool. Hygiene hypothesis. One of the reasons why they think about, it's good. Little kids should be around pets and animals, and now they're even like giving kids who, we should introduce like things like peanuts and peanut butter much earlier to kids so they don't develop peanut allergies and things. Anybody have a peanut allergy in here? You do? Is it bad or is it mild? It's bad. Okay, so I've been in schools where they don't allow anybody to have peanut butter or peanuts in the school. They don't sell peanuts. They don't make peanut butter jelly sandwiches. You could not bring a peanut butter. Somebody else in this classroom would not be allowed to bring a peanut butter jelly sandwich to the school because some kids have it so bad. Okay, so because it can kill you. I actually played a role where I helped cremate a young Canadian woman that was only, she was probably only like 20, 22 years old, but she died in a village right below our campsite in Central Africa. And her parents didn't have that much money, and she had a last will type thing. She letter that she carried with her that said, "Please just cremate me and take pictures and send them back to my parents." And so the Canadian ambassador was notified that this young woman had died, and he knew that we were in the forest near this village, and so he came out to us, and we went down, brought him down to the village, and built the funeral pyre and burned her body right there, and then sent it back to her parents, and she told them what happened and all that kind of stuff. But she ate some chicken that was cooked with peanut sauce. So if you ever go to Africa or you go to African restaurants, be careful because they use a lot of peanuts in their food. All right, so that's it. It was definitely an interesting experience, kind of like a big barbecue.